Hey folks, welcome back to the circulation unit. This is part number 4 of our unit. We are going to focus on blood itself. So this video we will go over components of blood, the different cells in blood, blood clotting, and then also blood types. So we'll preface this, see if you know what movie I'm talking about. I think of Ben Stiller. Nobody makes me bleed my own blood. See if you're a Ben Stiller fan. Know what movie that's from. Got a little Dexter scene here. Something look a little ominous. We'll call it just a bad nosebleed. Some blood splatter. So circulation part 4, looking at blood. So the different kind of blood cells that make up what we call blood. There are red blood cells. Their scientific name are erythrocytes. And in this picture, we are talking about these little guys right here. That's a sideways view. Remember, they kind of look like donuts without the middle cut out fully, just sort of sunken in the middle. So those are erythrocytes, red blood cells. We have thrombocytes, which are platelets, which are these little guys here. They're colored yellow in this picture, although the blood cells themselves, they actually have no color. This has just been artificially colored on a computer. So it's not like you'd have red cells and yellow cells and green cells. This picture was all black and white. The red blood cells have iron, which when exposed to oxygen, we know rust is kind of a reddish color. And blood is red when it is exposed to oxygen. There's lots of oxygen in blood. So that's why we call them red blood cells. But the platelets would have no real color. They're called thrombocytes, and we'll see that they're involved in blood clotting. And if you took Biology 11, I think you looked at immune response. You've also got white blood cells. That would be these guys here, again artificially colored. This one's light green. They are called leukocytes. So you do need to know those 3 terms, maybe more recognizing the 3 names than being able to like regurgitate them for me. But know if you see erythrocyte that we're talking about a red blood cell, thrombocytes are platelets, and leukocytes are white blood cells. There is a slide coming up, but leukocytes, that's leukemia. My cousins battled that, which is cancer of white blood cells. Something you definitely don't want to have, but that's where that leuko is the white blood cell prefix. So those are the 3 types of blood cells. In terms of blood, just generally, blood contains, actually the majority of your blood is not any of those 3 types of cells. It's actually mainly something called plasma. Plasma makes up just over half of your blood, 55%. And I think the human body contains somewhere between 7 and 8 liters of blood. So you're looking at 3.5, 4 liters of the fluid circulating in your body, in your blood vessels, is plasma. Plasma, so of that 55%, if we were to take out the plasma, it's mainly water. It's probably no surprise. And then the other 10% is dissolved substances, dissolved stuff. And what kind of stuff are we talking about? What kind of dissolved things are in plasma? Well, there's those 3 kinds of blood cells, erythrocytes, thrombocytes, and leukocytes. There we go. So red blood cells, white blood cells. Sometimes you'll see those shortened to RBC, WBC, and then your platelets involved in blood clotting. Blood plasma also contains, we had said the water, those 3 kinds of cells. There are blood proteins, so different proteins floating around in there. One of them is called albumin, which is something that your liver uses to maintain osmotic balance, to regulate how much water goes in or out of the blood. We don't need to worry too much about that, but just so you know. Fibrinogen is another one that's produced by your liver that's involved in blood clotting. We are going to look at the process of blood clotting, so we will see that one in more detail in a future slide in this video. There are gases. So dissolved oxygen, there's bicarbonate, there's CO2. We talked about that in the respiration unit. There are nutrients. We covered that in the circulation unit, right? The digestive system breaks down carbohydrates into glucose, proteins into amino acids, and fats into glycerols and fatty acids, and also some nucleotides too. So those will be floating around in your blood, dissolved in the plasma. There are wastes. Urea, which is that nitrogen byproduct when we break down proteins, and ammonia as well. There are vitamins. And if you take a multivitamin, I take actually half of a multivitamin every day. And if you take multivitamins, you probably have noticed this. My first urination of the day, I'll share this with you because we're all friends. My first urination of the day, if I take a multivitamin, after taking the multivitamin, I should say, my first urination is like hyper yellow. It's almost neon colored. And I think that's one of like the vitamin B12s or something like that. A lot of vitamins you take are water soluble, which means any extra just dissolves in your urine, and then you pee it out. And there's hormones, so things like testosterone if you're a male, maybe some estrogen if you're a female, and some other hormones in there too. And then there's salt. And remember, the salt is what's going to help maintain also that osmotic balance, so that you don't have too little water in your blood, but you don't have too much either. We have to have just the right amount. Otherwise, you can run into issues like we saw in that video before the break. I think Keller showed us that woman who drank all the water really, really quickly and somehow didn't vomit it up and ended up dying, which is crazy. That was a salt imbalance then, and water imbalance can run into trouble. So just enough salt in there to keep everything where it should be. Focusing in just quickly on the actual blood cells themselves. We won't spend too much time on this. I think we've chatted a lot about the red blood cells already. Of the red blood cells, white blood cells, and platelets, we have more red blood cells than anything else. So they are what we call a biconcave disc. So remember, they're kind of like a donut, but the middle's not quite punched out. Nice rounded edges so that they can slide through capillaries without catching. They don't have a nucleus. They actually do when they're produced. And then a lot of the organelles that we've talked about, they actually just dissolve and disappear. So these things are not really a functioning cell anymore. They house hemoglobin, and they house iron, and they house carbonic anhydrase as an enzyme that, remember, converts carbon dioxide into bicarbonate and H+. But they don't actually have a lot of functioning organelles at all. They're just kind of like a little raft for that stuff to roll around on. So there you go. Contains hemoglobin. We know that's the 4 seats for oxygen. Sits there in a tense state. When the first oxygen binds, it opens up into a relaxed state, and the other oxygens can bind on that cooperative binding of hemoglobin. We saw that last unit. Carbonic anhydrase, which is going to convert CO2 into bicarbonate and back the other way as well. And red blood cells, they last for about 120 days, so about a third of a year. Friction slowly breaks them down over time, just them bouncing around inside the blood vessels that we've chatted about in this unit. So friction breaks them down, and then they get recycled by your liver. There you go. Disposed of by the liver and recycled. And then your bone marrow in your long bones, so like your ribs, your femurs, your humerus. Want to hear a good joke about humerus? Just kidding. It's very humorous. Those are gonna to make the red blood cells. For somebody like myself, running all the time, running too much every day, my red blood cells actually probably don't last 120 days because they're circulating faster. There's more friction. They break down quicker. So I'm more at risk of being low iron, low red blood cell. So I have to be careful that I take either iron supplements or get enough red meat in my diet so that I have enough iron to have functional hemoglobin. White blood cells involved in immune response, so they're really big. They do contain nuclei, so they're kind of alive, and they fight infection. So they're kind of like the guards roaming around in your lymphatic system and in your blood, looking for bacteria and viruses that aren't supposed to be there. And a lot of them are phagocytic, which means, like this one is showing, they actually will send out these little projections and engulf things that aren't supposed to be there. And then usually they'll join up the virus or bacteria. Here we've got a little, is that a Diplobacillus bacteria it looks like. It engulfs it and then joins it up with something like a lysosome. Remember, that contains those digestive enzymes, breaks it down, and just kind of ejects or exocytoses, barfs out the dead, broken down virus or bacteria. So they're pretty cool. Again, the guards going around hunting things that shouldn't be there. Some produce antibodies. We're going to talk about antibodies in a little bit, so I won't mention that now. There's a slide that just shows some types of white blood cells and sort of what they're involved in. So there's histamine, if you've ever heard of that. Antihistamines are things you'll take to help out with things like allergies. Phagocytosis, when you engulf things to take them in and destroy them. Some involved in allergies, some involved in engulfing bacteria and pathogens. So kind of cool stuff. We again won't spend too much time on those, but just so you can see them. Platelets, we will talk about the clotting process in just a moment. They're very small, they're kind of irregular. You can see that these are all the platelets, these things in this picture, and then you can see the big red blood cell sort of sitting in behind that big biconcave disc. So their function is to form like a net-like structure for blood clotting to help us stop bleeding when we sever vessels of some kind. And kind of a cool Jeopardy fact, your body's going to make about 2 billion of these suckers every day. Huge number of platelets made in a 24-hour period. So there's a picture of bone marrow in the long bones. That is a femur, which is your sort of thigh bone. All of the blood cells, red blood cells, white blood cells, and platelets are made in the bone marrow of, again, I think it's your femur, your humerus, which would be like your bicep area, the upper portion of your arms, and then your ribs are the bones that contain marrow that are cranking out all of these blood cells. There's a slide, I'll let you read through that, just talking about leukemia, which is caused by cancer of your bone marrow and your blood potentially, and you end up with like too many white blood cells or white blood cells that aren't behaving properly. Unfortunately kills a lot of people and afflicts a lot of people every year, so not a good one. Okay, blood clotting. Junior, drink your blood before it clots. Ugh, kind of a gross thought. If you've ever had a nosebleed and then had a big blood clot in your nose and somehow sniffed, done one of those and sucked the blood clot back into your throat, you know that's kind of nasty. This is a bit of a process that you do need to know. There's a bunch of new terms that are going to show up here. There's a slide that's full of information. So I'll go through it, and then you're probably going to want to join me on Zoom so that we can chat about it in more detail and maybe help your brain work it out. So I'm going to plug that right now. Join me for a Zoom meeting. We should probably talk about blood clotting. So here it is. You damage a vessel, right? You get your, let's go with something nice like a paper cut. What happens is your blood starts leaving the blood vessel, and as you hit the rough edges of that paper cut, the platelets are damaged. You can see here, impact rubbing against the rough edge of the damaged vessel, and that's going to initiate the whole clotting process. So it's actually the rough edge of the cut that starts the blood clotting. So what happens is those damaged platelets release an enzyme called thromboplastin. So that's a term you're going to need to know. So thromboplastin is an enzyme that's released, and it binds with calcium, also that's in your blood. And again, you get calcium from lots of things like dairy in your diet. That thromboplastin enzyme and calcium are going to convert, so they're an enzyme, remember, so they're starting a chemical reaction. They convert something called prothrombin, which is a protein made in your liver. Prothrombin is not active, but the thromboplastin and the calcium convert the prothrombin into thrombin. So thrombin is an active form of an enzyme. It's kind of like how pepsinogen was converted to pepsin in your stomach by the hydrochloric acid. So there's an analogy. This is a little bit similar, where prothrombin doesn't really do anything. Thrombin is what we want. So the prothrombin, before functional thrombin, gets turned into thrombin by the thromboplastin and the calcium. Now it turns out that's not the end of the story. It's like this weird sort of snowballing cascade process. The thrombin then, it's an enzyme that's going to convert fibrinogen, so that's an inactive enzyme as well, into something called fibrin. And fibrin is cool. It's insoluble, which means it doesn't dissolve. So fibrinogen gets converted into fibrin, and the fibrin forms almost like a net-like structure that physically traps the red blood cells and the plasma and the white blood cells and other platelets from leaving the cut, from leaving the damaged area. So we actually have to make this net-like structure in the end. That's what a blood clot is. So it's thromboplastin and calcium converting prothrombin into thrombin, and then thrombin converts fibrinogen into fibrin. And the fibrin is the net-like structure. So that's what actually traps the blood cells. That's the clot, where it kind of gums up a little bit. So you're actually looking at, if you can see a blood clot, you're looking at fibrin, that insoluble protein. So there's some terms in there, right? You need to know, like the thromboplastin and calcium turn prothrombin into thrombin, and then thrombin converts fibrinogen into fibrin. So some new terminology. The more you sort of talk about it, the more you go through this, the better off you're going to be in terms of memorizing and knowing that process. So again, join me for the next Zoom meeting. Let's chat about it. There's a little picture. I'll let you pause and read through all of that so this video is not too long. We'll finish off this video with human blood types. This is pretty cool. Two terms you need to know are antigens and antibodies. So an antigen is a little protein on the surface of a red blood cell. And I should probably just put that little note in here. That might be easier than the note that I've got. So an antigen is a protein on the surface of a red blood cell. I'm just going to say RBC. Antibodies are little proteins that will bind to specific antigens and mark those cells for destruction. So an antibody binds to certain antigens. I'm going to go into the picture a little bit, sorry. And it marks that cell for destruction. So here is kind of an analogy for this. We'll see if it works for you. Again, hit me up on Zoom, because this is something else we'll want to chat about for sure on Zoom. An antigen is kind of like a gang tattoo or a prison tattoo, where if you have a certain prison tattoo, then some people in the prison will look at that tattoo and go, no, you're okay. You're one of us. We recognize that tattoo. We like it. We tolerate it. You come hang out with us. So your body, for example, I'm type B blood. That means that my red blood cells have antigen B on them. And my body recognizes that as being okay. So it's kind of like all of my red blood cells have the antigen B, the B tattoo on them. And my body looks at that and goes, yep, that's okay. I like tattoo B. But there are other blood types like blood type O and blood type A. Those are different tattoos. And if you have one sort of gang tattoo in prison and you go to a different area where they have different tattoos, they're not going to like yours. So my body tolerates tattoo or type B, but it does not like type A. So if I were to have a blood transfusion, if I were to have blood added to my body that had antigen A, the A tattoo, if we stick with that analogy, then my body would go, whoa, 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 what is this? We do not like that tattoo, right? We don't recognize it. That's not our kind. And it would produce all of these antibodies, which would bind to the type A blood, and then my body would start attacking and killing the type A blood. Just like if you've got the wrong prison tattoo or gang tattoo and you go into a different area of that prison, you might get yourself beat up, right? Because people are going to look and go, no, 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 we don't tolerate that one here. And they're going to mark you for destruction. And you might get yourself into trouble. So that's how this sort of like antigen and antibody thing works. Antibodies mark things for destruction that your body doesn't recognize. Things that are foreign that it doesn't like. And antigens are things that your body marks and tolerates. They're like prison or gang tattoos. Kind of a harsh analogy maybe, but it works. So the blood types, maybe you've heard of this, universal donor. Got a little alien in there. Sorry. Bad science cartoons. So the red blood cells have antigens on them. And depending on their antigens, their prison tattoos, we call them by different names. So people with blood type A have antigen or tattoo A. So that means when you get your blood type, you get one copy of a blood type from mom, one copy of a blood type from dad. So somebody who's type A blood could be AA, where they got a capital A from, or tattoo A from mom and tattoo A from dad. So they've just got tattoo A's on their red blood cells. Or maybe they got tattoo A from mom, but an O from dad. O is actually the absence of an antigen. So somebody who's AA, let's say that it looks like this, their red blood cells. They have A's, and then the O actually means you have like nothing there, right? So there's somebody who's AO, where they've got some A's on their red blood cells and then maybe some little sticks with nothing, some missing gang tattoo spots, right? They're not fully tattied. Whereas somebody who is AA would have lots of A tattoos. And it doesn't matter whether you've got like fully tattied with your A's or just some of them. You will be tolerated by or recognized as A. So you've got gang tattoo A, regardless of whether you've got 3 or 6 or 1,200 or 20,000. It doesn't matter. So that would be blood type A. Then there's blood type B, which means you've got antigen B on your blood cells. Maybe you're like really, really full of B's because your genotype is BB. Or maybe you've got B's and then some empty spaces, some O's. Remember, O's are just empty. So a B person might look like, sorry, this looks like a white blood cell now, but we're still talking red blood cells. Here's somebody that's type B. This is myself. So maybe you've like got just a few B tattoos. Maybe you've got lots of B tattoos. It doesn't really matter. You have type B tattoos. There are some people, if one of their parents is a type A and one of their parents is a type B, then they actually end up with both tattoos on their red blood cells. So their red blood cells have A's and B's. And what's kind of cool about that, A, B, A, B, A, B, is that you can go like anywhere in the prison yard that you want, and people are going to tolerate you because they recognize one of your tattoos. So it's not like that they look at the other one. So my body, who's type B, only tolerates B. People who are AB, they tolerate A's and B's. So you could give those people type A blood and their body goes, you got the type A tattoo? Yeah, you're okay. Or you could give them my blood, type B blood, and they'd look at it and say, their body would say, you have type B tattoo? Yeah, okay, we tolerate the type B tattoo as well. We recognize that one. So people who are AB will tolerate anybody with tattoo A, or anybody with tattoo antigen B, or anybody with both. As long as they see an A or a B, they're happy. And then there's blood type O, which is no antigen at all. These people, if you have no antigen on your red blood cells at all, then you actually look like this. We don't put an O because it's actually the lack of a tattoo. So these people almost have no tattoo. So they're tolerated by A's and B's and AB's. Everybody likes type O because they have no gang tattoos at all. They have no gang affiliation. So the gangs don't get angry at you if you're not affiliated with anybody. So type O blood, you can give that to people who are type A, or you can give it to people who are type B, or you can give it to people who are type AB. They're called the universal donor because their red blood cells have no markings whatsoever. And so everybody looks and goes, yeah, you're like Switzerland. You can hang out here. You're fine. It's only when you see those tattoos that you don't like that you develop those antibodies and you start to attack that. There's also this whole plus-minus thing. Here's a nice little picture that shows what it might look like. So people who are type A might have like the round antigen. Type B, this little diamond-shaped antigen. AB has both. And O type means you have none. Antibodies show you who you're going to attack. So somebody who's type A tolerates A, but you can see they've got these antibodies that will latch on to anybody who's got a B, and they're going to attack it. And on the flip side, people who are B, so this is myself, I'm blood type B, so I have these special antibodies that are on the hunt for tattoo A. You can see that these are round and they fit on there really nicely. They mark type A blood for destruction. People who are AB, they don't want to attack anybody. They tolerate everyone because they recognize all gang tattoos, right? Anybody can hang out there. And then there's poor type O. They can donate their blood to anybody, but if they receive type A blood, they're going to attack it because they're not used to it. And if they receive type B blood, they've got antibodies to attack that as well because they're not used to it. So O can donate to anybody else, but it will attack any other kind of blood. So they can only take their own kind of blood. Rh factor is something called the rhesus factor, named after the, I'm probably saying it wrong, but rhesus monkey. Here's a random Jeopardy fact. So it was first discovered in these rhesus monkeys, and now we just call it Rh factor. They're kind of like another prison tattoo as well. So somebody who is Rh positive, so my blood, remember I said I'm type B, to go a step further, I'm actually B positive. So that means that I have, here's my red blood cells, I have type B on my red blood cells, and I also have this positive Rh factor. It's like an additional tattoo. So my body looks and goes, okay, I'm used to B's, and I'm used to having this positive tattoo. So it's used to those two things. If it sees A tattoos, it attacks. If it sees O-type blood, that's okay, because O-type, remember, is no tattoo at all. The other thing that's interesting here is a person who is Rh negative. So some people might be like A negative blood. Negative means like no tattoo. So it's kind of like O-type blood. So for me being B positive, I can actually accept B negative blood because a B negative looks like this. It means it's got B's and then it's got no tattoo to mark the plus or minus. So it's not like that. You don't get a negative tattoo. An Rh negative means you don't have any special Rh tattoo. So my body tolerates that, right? Because it's looking and it tolerates the positive tattoo, and it also tolerates the lack of a tattoo. It doesn't mind that. Somebody who is B negative, I'm not B negative, but somebody who's B negative would attack the positive tattoo because it looks and it goes, oh, I'm not used to that positive tattoo. Attack it. Who is that? Right? Some other gang. But if it sees no positive tattoo, then it's okay with that. So somebody who's Rh positive, they accept positive tattoos and negative tattoos because that's the lack of a tattoo. That means there is no Rh tattoo. But people who are Rh negative can only accept negative blood. They can't accept positive because they would recognize that positive right there as a bad tattoo, some rival gang, and they would go to attack it. So here's some random picture. It doesn't even really make sense. I don't like it, so we'll ignore it. There is within your circulation assignments a document that talks about blood types and who can accept from who and who would you attack. You should give that a try. And it's usually something that a lot of students, it takes a minute for them to wrap their head around how that works. So I won't make a video of it. I'm going to say you should know how it works. It's really good to know just generally how blood typing works and what you can accept and not accept. So why don't you join me for a Zoom meeting, either Tuesday afternoon or Thursday morning, and we can go over that worksheet together and talk about this whole like A, B, O, Rh positive, Rh negative, because it really will be beneficial and be a lot easier for you if we talk about it in real time with a worksheet. So join me for a Zoom meeting or fire me an email and we can schedule our own little Zoom meeting because it'll make your life much easier. I hope to chat with you soon. Have fun thinking about blood.